Good afternoon, everyone. I'm Simon. I'm broadcasting to you from our Life Hub in Lyon. I'm really pleased to be here today, and I'm really pleased that you're here online with us. Just a little point about the current situation in France. We're at work today and we're all obliged to wear masks in the Life Hub, so you'll see us all wearing the masks. It's for our safety and it's because we take safety seriously. We've got a fantastic lineup for you today. Just to give you a view on the agenda, we'll start with a short introduction. We'll learn a little bit about open innovation at Bayer from Bernard and Catherine. I'll go quickly into the challenges and what the challenges were when we posted the Hello Tomorrow Startup Challenge back in the summer. And then we'll dive straight into the exciting part of the different pitches that we've got planned. We've got 6 pitches planned for you today. Each pitch will last about 6 minutes, and then there'll be 5 minutes for questions afterwards. So please don't hesitate if you're watching this from video. Please don't hesitate to ask your questions in chat. And we're really lucky today. We've got Nelly as our moderator, who will be moderating the questions and the pitches. Another exciting part on the agenda is we plan to have some voting. So at the end of the pitches, we'll flash up on the screen, and we'll put the details in the chat as well. But on the screen, you'll have a QR code and you'll have some information on how you can log in. And you in the audience today, you get to choose as well. You get to choose for your favorite startup, and you can vote for that, along with our judges that we have lined up today, who will also be deliberating live today in the back room and online to select our favorite startup. And then stay tuned and stay on board until the end, because right at the end, we'll have the winner's announcement, and we'll have a short ceremony and a short look back at the event and announce the winners from the voting. So I think that's from the introduction, that's everything. So let's go and let's learn, let's start and learn a bit more about open innovation at Bayer Crop Science. Thank you.
Hello, Catherine Sirven. I'm responsible for digital transformation in research, and I'm so happy to be there today as part of the Life Up team also. Today we welcome Bernard Leroux. He's Senior Vice President for R&D for cereal, rice, and oilseed rape, and I'm really happy to welcome him. And my first question will be on his view on innovation. What does it mean for you, Bernard?
Thank you. Good afternoon, everyone. During my more than 35 years trying to do R&D in the world that I am into, I had a lot of view on what is innovation really. For me, at the end, it comes to looking at a problem from a very different angle to be able to find totally new solution. If I take as a layman example the world of automobile, to actually change your view from improving even further the motor, which is always a view, and to try to recover the energy you lose when you are decelerating, coming to hybrids is a crucial way of looking at things differently and bringing a total innovation with the hybrid car. To decide that basically the driver is not anymore a fundamental part of driving a car is also a major step into innovation. In agriculture, to think that insects, by the fact they are mobile, could come to the bait that is going to kill them or actually stop them reproducing has led to major advance in the way we are controlling pests, both in the agricultural world through either transgenic plants or through seed treatment or even through baits, but also in the fight against malaria, with the fact that when we invented Bennett, we actually used the human being sneak peek under the net as that bait, and we were able to control mosquito much more effectively. So those things are example of how looking at things differently open new ideas for solving the problem.
So through your examples, we have really seen that agriculture is a key topic here. And for you, what does it mean, innovation today for agriculture. So what are the trends you see coming and how you deal with that?
So for us, we are in front of a major challenge, which actually not for us only is a challenge for humanity, is the fact that the growing population requiring that we are able to feed people, linked also to the fact that we cannot further degrade the biodiversity that is still existing on the planet, and the shrinking resources that are there. Additionally to the fact that climate is not helping. Climate is a fundamental element in agriculture and is today changing to such a way that farmers are being challenged very regularly for this. This creates a situation where more than ever innovation is needed to actually attack the problem. That innovation doesn't come only from the classical way, and I would say even further it doesn't come from the classical way. We have to change the way we look at things. And as such, we see the advance of digital technology. We see the advent of better harnessing the knowledge in biology as a way to actually improve the situation.
Okay, so I come back to the final questions. This is for Bayer. Innovation is really key in the center of our research and way of thinking, but maybe you have some comments and examples about innovation in Bayer for agriculture?
We are actually working on a number of things. Today, we have a classical innovation, I would say, improved products that are actually going to substitute older products that I would say are either not effective enough anymore or actually present a profile that is not so favorable for the environment. We are actually continuing to find new solutions for resistance but on top of those classical elements, we have actually started to work on biological project harnessing the knowledge we have on biology of microbes to actually improve them and offer another area of solution. We are looking at digital solution to try to better position product, help the farmer make better his decision, and help reduce the use of inputs for a better output. We are actually also using those technology to improve the way we do things internally. And finally, we are looking outside with other companies as novel way to control pests. For example, we are working with people to actually develop Wolbachia type of mosquito. We are working at different ways to use genetic engineering to actually modify the way the pest population can actually work. So there is a number of things that are linked to better understanding of biology, better ability to compute all the knowledge we have that are allowing us to look at things a very different way. And in this, the collaboration with a broad array of scientists all over the world is fundamental. If we didn't have this, I think we would not be able to go fast enough in finding the solution.
Yes, and of course with that, we will see a concrete example of this open innovation, and I transition back to Simon so that we can talk about this challenge where you will see the pitch from external companies today.
Yes, so thank you, Bernard and Catherine, for that short introduction on open innovation at Bayer Crop Science. And as you said, Catherine, we're going to use today to showcase one concrete example. Back in the summer, we contacted Hello Tomorrow, and we worked with them to organize an international startup call. I'm going to show the questions that we asked here. So we organized it with Hello Tomorrow. So obviously we faced with many problems, and this isn't an exhaustive list shown on the screen of all the problems we have. These are the ones that we've chosen that are high priority. And you'll see in them problems and questions around sustainability. You'll see in these questions, you'll see questions and problems around early pest detection, leading to perhaps a yield enhancement or yield maintenance. And you'll also see subjects covering digital in silico technology So digital toxicology or in silico toxicology, an important part of our pipeline and how we can drive our pipeline forward, and using new technologies that weren't invented in Bayer and using partners like Hello Tomorrow has been for us absolutely fantastic. And I think on the next slide, you'll see just some ideas of what happened in our challenge. So here in the challenge we ran throughout the summer with Hello Tomorrow, we had a total of 124 applicants based on the 3 pillars of the questions that we asked. And those applicants were really, truly global. I mean, we had about 1/3 coming from the EU, so companies or startups with their headquarters based in the European Union, and then about 20% or 1/5 are coming from North America, so startups with their headquarters in North America, and another 1/5 are with their headquarters in Asia. So we managed to get with Hello Tomorrow a truly global reach of really fantastic and interesting startups, of which we're going to hear only from 6 finalists today. So for me, a big thanks in advance to all the finalists for their time and their efforts, and then being here being here with us today for the event. One other point I want to make is we don't only have the judges online. We don't only have the startups online for the pitches. We also have a group of esteemed and experienced judges who are going to judge the startups on a number of pillars, including the technology and including their sustainability elements, which you'll see is a running theme throughout the presentations. So in the judges group, we have Sasha and Melanie just reading left to right. We have Arlene, we have Anna, we have Phil, and we have Bernie here with us. And we're really looking forward to seeing which startups they choose in the finals today. As I said, we'll have the announcement at the end of the event. And also, as I said, if you can remember, at the end of the pitches, there'll be a QR code and a website address on the screen, which will also go into the chat. And you'll be able to also cast your vote for your favorite startup, which we'll announce as well at the end of the event. So the startups will be 6-minute pitches. We've got Nelly ready and waiting, I think, for the question and answers. And I can already see that Yoav has turned his camera on. Yoav, how are you doing? Are you ready?
Yes, I am ready.
Fantastic, Yoav. I'll control the slides from my side. So if you could just say next slide, please, for the next slide. Yoav, if you're ready, your 6 minutes, they start now.
All right. Thank you. Thank you, Simon. Thank you, Hello Tomorrow and Bayer. Happy to be here. So let's start with the first slide, please. Thank you. So hello, everyone. My name is Yoav Adomi. I'm co-founder and CEO at Agrosage based in Tel Aviv. In the next 6 minutes, I'll explain how we help greenhouse farmers become winners in their constant battle against pests and diseases. Next slide, please. So today, plant protection procedures haven't been changed really for decades, and they're pretty much based and carried out by a professional, a pest and disease specialist. Now, this professional resource, as you all know, availability is very much limited. And this is why the inspection is not very frequent and not frequent enough, every 7 to 14 days or so. B, statistic, hence many findings are often missed. C, there is no early detection whatsoever. And lastly, there is no use of big data, not for the inspection, but not for the treatment recommendation. Next slide, please. The result is that the farmers today are being defeated by pests and diseases, and they are losing because there is an extensive use of plant protection materials. They are breaching MRL. Plant protection materials are being actually broken, as you're all well aware of, because pests are developing resistance. There is a meaningful environmental impact on both soil and crop itself. And lastly, they are suffering from a significant yield loss, which is, of course, very, very important. Next slide, please. So Agrosage is here to make the farmer a winner. We are doing this by disrupting the entire plant protection processes, from sampling to treatment protocol. We are digitizing the professional knowledge of the pest and disease specialist, and we're making it accessible to any farm worker, making, if you want, any farm worker the world's best pest inspector. We are putting our emphasis on early detection by leveraging AI technology and having a digital specialist resource available at all times. And lastly, our adaptive scouting and adaptive treatment methodology supports both early detection and the reduction of plant protection materials. Next slide. Here we will see and go over quickly of our solution architecture, which is, by the way, already being field tested for more than 8 months. It all starts with a smart and adaptive scouting. No professional is needed. The scouting device can be a simple smartphone at stage 1 or a wearable like smart glasses with Agrosage 2.0, and later on an autonomous robotic scout. Next slide. All of them are providing foliage photos that are analyzed on the fly by our proprietary 5 AI algorithms. Next slide, please. In this example, you can see the resolution of the analysis. So we are focusing mainly, but not only, on the bottom side of the leaf, which is crucial for early detection, taken from different heights of the plants, and according to the methodology, separating between pests and beneficials, classifying by type, life cycle, and counting on leaf. We are identifying different phenomena, and we're setting infestation level, local and global. Next slide, please. Lastly, we are providing an adaptive and focused treatment recommendation, which takes into consideration all of the relevant data like infestation level, conflicting treatment, weather forecast, harvesting and local regulation. And needless to say, for a human, even the most professional one, it's almost impossible to digest all of these important criteria for an optimized treatment protocol. Next slide. So the farmer has access to an advanced dashboard where you can track the infestation spots in the greenhouse, follow trends, filter by pest and disease, generate reports, and approve the treatment recommendation. Next slide, please. To conclude, the 3 pillars of our solutions are 1, systematic and frequent inspection, 2, early detection, and 3, adaptive treatment. All leads to the reduction of plant protection material use and yield increasement. Next slide, please. As for sustainability, we are looking at 3 main points. 1, and I think it's very clear, the reduction of plant protection materials, which is driven from the early detection and focused treatment, meeting the MRL requirements, and healthier soil and yield. Next slide, please. So this is the evolution of the domain as we see it. The current solution belongs to the past. It requires a professional. There's no early detection. Today, we are already offering the mobile-based solution, accessible, scalable, and no professional is needed and supported by AI. Our 2.0 solution is a wearable based on smart glasses. It will enable fast, hands-free scanning for bigger-sized farms. And lastly, Agrosage 3.0, is our vision for autonomous robot that will provide full cover, not only for pests and diseases, but also nutrition, pollination, and more. Last slide, please. So today, as you can see, we are in orange belt fighters. Collaboration with Bayer will help us become Bruce Lee. We see 3 main objectives for such relationship with Bayer. 1, Bayer is a strategic investor, investing between $2 to $3 million. 2, Bayer is a strategic partner, helping us to shape the product, reach product market fit. And lastly, go-to-market assistance. With Bayer, we can meet the markets at the earliest, decrease the time to market, and use the right distribution channels. This is it, guys. Thank you very much. Thank you, Bayer.
Thank you, Yoav. Let's now switch to the Q&A session. So what makes your product stand out in the market, and how would you invest funding from Bayer to improve it?
Okay, as for the product in the market and the differentiation, I think that we are one of the only startups, maybe the only one, that takes the entire, I'd say, process from sampling, adaptive sampling, very important, supported by AI, through the analysis, AI analysis, and lastly, to provide an adaptive treatment recommendation, which differs from any other player in the market. They take into consideration all of the variables that I've mentioned and using IPM methodology. So that's something that really differs from the entire market. And what was the second part of the question? I'm sorry.
How would you invest funding from Bayer to improve it?
Thank you. So investment from Bayer will be, I'd say, separated into two. One is finalizing the development today, especially the AI development. At the moment, we support not all crops and not all diseases, but the most popular ones. And we need to build a scalable and to cater to more crops like cucumbers, peppers, not only tomatoes and others. So that's one. And second, we will also be focusing on go-to-market and investigating on developing the right distribution channels. Again, with Bayer, we think that we can reduce the time to market and make it much more efficient.
So how much has your technology been tested in the field?
So we are already post-pilot. We already did about 8 months of extensive field testing, testing I'd say the three main pillars of the solution. One is the adaptive scouting, making sure that any farm worker with not being a specialist can actually perform the scanning according to our methodology. So we already did that. And it is not only that we scan at least as good as a pest specialist, but most of the time we even find better results. We show better results. So that's one. Second, proving that we can, based on this scan, we can actually provide a treatment recommendation that also is in correlation with the status on ground. And lastly, we also validated the AI algorithm, showing that we can, by taking photo, an RGB photo, we can count the pests on leaf. We can detect them by type. We can separate between beneficials and pests, and we can also track different phenomena like powdery mildew, mold of all kinds, yellowing, and so much more. So that's already been validated, and the entire, I'd say, UI and dashboard and end-to-end product is already developed and tested by farmers.
Okay, previous questions were from the jury. Now I'm taking a question from the audience. Is it possible to extend your system to pathogen detection?
Yes, it is possible. We did already some initial testing about that. So the answer is definitely yes.
Okay. Maybe a last question. What will be your commercial argument to convince or convert traditional farmers to use your product?
So I think that not only that I won't be the only one convincing them, meaning that the market will convince them. And by saying that, I mean that we're seeing the trends, both the trends from a regulatory perspective, but also consumer trend of consuming better crop, reducing, not consuming a crop that have been heavily treated. And that brings the farmers, even today, to use greener solutions. And when using greener solution, you have to be focusing on early detection. Otherwise, you won't be a player in the market because the soft materials are not as effective as the heavy ones. And this is why digital solutions are a must. By the way, especially in these COVID-19 days that we're seeing that specialists are not really available at all time and everywhere.
Okay, Yoav. Well, thank you. I think we can finish now the Q&A session so that we can have time to listen to the other startups. So you have presented Agrosage. Thank you for the pitch and Q&A session. Now,
thank you very much.
Thank you. Now let's discover Ananda, a startup led by Margaret. Margaret, are you ready?
Yes.
Okay, so your 6 minutes start right now.
Bonjour. Thank you so much for organizing this great event. It's a fantastic opportunity to join Bayer in this mission for sustainable solutions for in silico toxicology. My name is Margaret Magdesian, and I'm the founder of Ananda Devices. Next slide, please. The limiting factor for in silico toxicology today is good quality data. the vast majority of toxicology data is based on animal data and can very poorly predict the toxicity of compounds to humans. Let's take neurotoxicity as an example. 20% of the world population suffers from neurological diseases, and the numbers are increasing every year, especially now with COVID. However, over 99.6% of all the drugs targeting neurological diseases tested in animals fail in human clinical trials. Why is that so bad? Because animals do not naturally develop the most common neurological diseases that affect humans, such as Alzheimer's disease, Parkinson, multiple sclerosis. But the toxicology database is based on animal models. So we're testing if a drug is efficient and safe for humans in a model that will never develop a neurological disease. Even worse, we women have much higher chances of developing neurological diseases. However, we represent less than 25% of all the databases because the vast majority of the safety tests are performed in animals and in male subjects. We have to change that. Please, next slide. Toxicology databases are biased towards male and animal data. And if in silico platforms will be fed with current data, the bias will just amplify and the risk for new products will only increase. Next slide. To really predict how compounds are affecting the population, we need human data and we need human-centric solutions. And that's why I'm here today. I'm bringing tools and I'm inviting you to help us change that. Please, next slide. I have developed proprietary technology to organize neuronal networks in vitro in miniaturized structures 60 times faster than they grow in vivo. What we do is we collect human data, and this is a scalable platform compatible with standard automation equipment. How does it work? So first, we offer our proprietary microplates to organize these mini neuronal networks derived from blood samples from male and female samples. Next, we add a certain compound. Next, we put this in a standard automation equipment, and in 20 minutes, we can collect data from 3,000 human neurons, enabling automation and correlation with the clinical and genetic data of this patient. It reduces costs, accelerate testing, and enable automation. We can get big data faster. Please, next slide. The main advantage of our technology is speed, because everything is organized, miniaturized, and we develop this technology to grow the neurons much faster, we can get data much faster. Because everything is miniaturized, we use much less volume, and then we reduce toxic waste by 90% doing testing. We also significantly reduce the use of animals along the whole path of product development. Our technology, if we use human neurons from the beginning, we don't even need to use animal data. We can directly move to the next stage of the trials. And most importantly, we get gender equality in health, and it's about time. Please, next slide. So this is a fantastic opportunity to to partner with us for human-centric, sustainable solutions. Together, we can develop better and safer products for the population faster. We can increase environmental safety. We can reduce the gender gap in healthcare. And most important, for future generations, we can build a database that represents our society, including all colors and all shades of humans. Please, next slide. We are here today inviting you to lead us from the past to the future, from cell lines and animals to human cells and reproducible results, from manual subjective technology to automation and big data, from single platforms to high throughput screening. Please, Bayer, let's do this together. Thank you. I'm open to questions now.
Perfect. You've been very sharp, very clear. Okay, so now I have a few questions already in the system for you. My first one would be, do you plan to extend your technology to other tissues?
Yes, indeed. We already signed a few contracts with other companies, more specific in the cosmetic field, where the use of animals is really restricted. And we have been using neurons together with innervation technologies to innervate different tissues.
Okay, great. How does your approach correlate with in vivo data, animal clinical data if available?
Well, that's what we need to validate today, right? The use of stem cells. So we use stem cells from human blood. The use of stem cells has started in the past 10 years. There's plenty of studies now using different models of humans. But the major advantage, we have a recent paper showing this, is that for the first time, we can isolate the neurons that are in the central nervous system to the neurons in the skin. And then when we test it, we get the effects much closer to what happens in humans than what's happening in the animal.
Okay. Another question, can your models be adapted to the needs of agrochemical companies as the safety testing strategies are fundamentally different, in particular, the absence of clinical data?
Yes, that's why we're here today. Our major goal is to validate our technology for the food industry and show that we can become the standard for the gold standard technology for in vitro technology, for in vitro testing of safety.
Okay. What is the advantage of other tissue or organ on the chip technologies or providers?
It's time and throughput. So there's a lot of people building very complex models that take 6 months to several months to produce, a mini brain, for instance. It takes at least 4 to 6 months to produce those, and it's very hard to scale. And it's very hard to image because, it's a 3D structure. Things can get very complicated. What we offer is a 2D structure with throughput. As I said, this is our product. We have 3,000 neurons in one of those, and it's standard, can be read in any type of standard equipment the company already has. All we need to do is just get the data and validate with the current database.
Okay, we have time for additional question from the audience. So is your technology applicable with other cell type?
Yes, we have used also in cancer, and we have over 20 years of experience in neuroscience, and I have developed this technology with McGill University at the Montreal Neurological Hospital. So most of our data is really on neurons, but we have already applied to skin, we have already applied to cancer, and in parasitology as well.
Okay. Will you take the world population into account, for example, based on different nutrition habits?
Yes, this is a very important question because that's what we want to change now. If you take directly the cells from a patient, not only you can focus on cells from men and women, but also we can take cells from people that have diabetes and not diabetic patients and test if a compound has a different effect on them.
Okay, so I will take a last question from the audience. You will have about 1 minute and 30 seconds to answer it concretely. What are you measuring and which aspects of neurotoxicity are you trying to predict and how do you make the connection? So it's 3 questions in one, sorry for that.
If you can show slide number 10, please. I have an extra slide, it's number 10, and this shows all the applications of our technology. And basically, so I don't know how long it takes to show it, but we can use the technology for compound screening or repurposing to investigate the mechanism of action, for toxicity testing, disease modeling, or even for personalized patient responder analysis. Slide number 10, if you don't see it, is a slide from the neurons of a cancer patient in focus.
Okay, I don't know if the technology will be on the side. Maybe.
It's okay.
Okay.
Yeah, there was some extra slides. If they're not there, it's fine. I'll be happy to show you or send the slide to the person who asked. Come to our website. We have it there.
Okay, well, Margaret, thank you. You were presenting Ananda. You received a lot of questions from the audience. Sorry for those that we haven't been able to handle, but we are taken by the time. We will definitely try to share that with the startups so that you can get the answer. Now, I think we can give a round of applause for to Margaret, and I will move now to the next startup. Let's discover Fotenix. It's a startup that Charles will introduce. Charles, are you ready for the pitch?
I certainly am, Nelly. Thank you very much.
Perfect. So your 6 minutes starts right now.
Good afternoon. My name's Charles Vase, and I co-founded Fotenix back in 2018 to address the deficit between what was available and what was possible to support crop scouting. This is why Fotenix concentrates on being an ability provider, enabling smart operation of agricultural machinery in real time. We act as a conduit between the plant and the equipment used to cultivate it, and we focus on embedding our diagnostic capability and empowering the producers that use it. Next slide. The real question comes here is about precision agriculture and what is precision? Agriculture in this sense is an ability to adapt in dynamic changes in a production process and to optimize the resources and minimize the waste, but on a per plant basis. There's been great advances in aerial technology, recording variation in fields, but the benefits are hampered by poor results and delays in relaying this insight. The problem between resolution distilling through to timing, which is so important in the agricultural setting. What is the matter if you have the best pesticide in the world, if it's applied at the wrong time or in the wrong place? Next slide, please. At Fotenix, we designed tailored hardware specific to plant material. We use cameras and LEDs of different colors deployed onto the plants to detect changes in the status. For the technical ones around you, this is a method called 3D multispectral imaging, and we create a digital twin of the plant, recording its information to do with content, which you can see in the image on the left, and also the 3D shape, which you can see in the background. The example you see here is an oilseed rape plant run through trials at the P3 Centre in Sheffield. We were able to detect light leaf spot within this plant in half the time it took for visual inspection. And this was during its pre-symptomatic phase, i.e. there was no visible symptoms to the human eye or a standard camera. This is all powered by a cloud AI platform, which allows edge deployment of our machine learning algorithms to run in field or in the lab in real time, but also allows us to sync devices so that one system can inform the entire fleet and vice versa. So there is compounded information that grows as more of our devices are out there and deployed. Next slide, please. We have used this system already in a number of applications through our Delta crop information platform. Firstly, in the lab environment for trials such as breeding and individual potted plants to create full digitized crop trials. We've also deployed it on board our customers' infrastructure of robotics, in particular Saga Robotics in polytunnels and strawberry farming, and we are currently working to integrate the capability inside vertical farms, which would allow a fully automated production process. This shows the different scalability and different areas of which the technology can be applied. Next slide, please. But why does this matter? Well, what is the benefit? Particularly varietal selection. With the example I gave you with oilseed rape, we were able to tell not only what the percentage of infection was on each of the plants, but also where that was spatially distributed, and to do with the bottom leaves of the plant and the top leaves of the plant. And we were able to identify one variety that had 30% less infection evolution than the others. This then trickles down into do with pesticide applications. Such a system would be able to inform not only the timing, but also the distribution. Imagine what that would do in a model as we move from euros per hectare from 2 euros per hectare from euros per litre. In terms of the bottom line, we are end user centric and believe that the importance is that the growers are able to improve soil health through data driven stewardship, but it also enable it in a profitable manner so that they are incentivized to use such technologies and such capabilities. Next slide, please. But who are we? At Fotenix, we're a mixture of teams for those who are experienced in both the agriculture and the technical area. We have a mixture from the agricultural side, from the likes of Tesco's, ADAS, Syngenta, and Velcor, right through to technology with experts from Leica, Broadcom, and those at the forefront of AI. In terms of myself, I have a doctorate in agricultural diagnostics, and I've been building camera systems since the start. The point of the team is that we're customer focused and market aimed. What we've done is found a problem and designed a solution to it rather than the other way, which sets us apart with some technology providers.
1 minute.
Next slide, please. Our interest is that in one agriculture, and this is our call to action with the likes of the behemoth that is Bayer. We believe that these problems within the sector will only be addressed through an open innovation approach with collaboration across the supply chain. And we believe the same in our business model, spreading our cost and our value both to the users and those that benefit from the information it could provide. We would like to continue our journey into the breeding sector and deploy our Delta detection system, in particular with disease detection, with Bayer in terms of their breeding trials with our crop diagnostic platform. Next slide, please. At Fotenix, we believe in providing diagnostics with data-enabled decisions. If you're interested in anything I've mentioned today, please get in touch, either through our website or through the details on board. We've been Fotenix, and I've been Charles Vase. Thank you very much.
Thank you, Charles. So now here are some questions from the jury. Is the technology able to work under outside conditions? And what is the lag time between data acquisition and final analysis?
That's a great question. So the trials that have run outside have run under two conditions. They've either run underneath a spray hood, so in the shade, or on the automated systems, they have run at night, where we get the most improvements in operation. In terms of the lag time, it does depend on the application. So we provide basically a video stream in real time for things that are able to detect, but there are some things that take slightly more processing, and then we provide it after it's been processed. So that could be in the case of 4 to 6 hours.
Okay. So what makes your product stand out in the market, and how would you invest funding from Bayer to improve it?
Our product stands up in the market both with our business model in the way that we're looking to be an ability provider to sell an item. We work with our customers. We're not looking to sell kit to growers or to agronomists. We're looking to integrate as an OEM supplier with machinery providers. And that seems to create quite a lot of traction, and why you'll find a number of our devices on board some of the machinery that's already out there. How we would use an investment from Bayer? Well, the importance for us right now is scaling. We went from demonstrating the technology over the past 3 years, and now we're looking to deploy it over a number of areas to be able to test and develop our cloud infrastructure system, and to be able to show the real benefit of how it works commercially with all the partners involved across the supply chain.
Okay, so your solution seems to be mostly focused for controlled environment or greenhouse application. Can it be expanded to field use?
That's a great implication. So yes, we've used it in controlled environments initially, which was the sector where we had the most value added and where there was technology where we could mount across. We do have trials in the field right now, and our only limit is to do with what machinery is in there. So if there are any machinery providers out there looking to test this capabilities, we are able to deploy it and tailor it to their needs.
Okay. What are the key technical milestones to achieve with your current prototype?
So the prototype that I demonstrate to you is commercially available. It's been commercially available and sold since March. The technical milestones to do now have been actually tied with some of your previous questions in how we would take it from a crop information platform into an online system at a lower cost that would run in a field over larger areas to address the markets that are we operate in the UK market where the farms are relatively small compared to some areas. And it's a question of scalability because the information is very large. That's our most technical milestone, and we'll be complete within the next 12 months. Okay.
Could you tell us more about your go-to-market strategy?
Yes, certainly. So our go-to-market strategy is to be an OEM supplier to machinery providers. We mostly focus with controlled environment and robotic providers at the moment. but we're also looking at deploying our technology as a retrofit on equipment. The reason for this is we believe in focusing on what we are good at, and what we are good at is providing information and translating information from the plant. There are lots of good service providers and data platform providers that we are working in an open innovation way to integrate with, to ensure that growers have a selection of devices that have compatibility, which is something which is not traditionally seen in the sector with siloed development.
Okay, so here's a technical question from the audience this time. Would it be possible to place the sensors on a drone?
We get this question an awful lot, and in answer, it is possible, but it also depends on why you would want to do that. We really focus on being able to enhance machinery. So if you're using spot application, we tell your device where to apply at the time of application, which is when it's needed. The use of drones are great for telling you something is there, but the level of information is not there and won't be for some time. So the question is technically possible, but commercially would not make a huge amount of sense in today's climate.
Okay, so one last question. If your company ends up partnering with Bayer, what would you do during the first 3 months to achieve your goals?
I think the first thing we would do is we would deliver a number of our Delta information platforms and get the crop pathologists to be working with those to show the information, and then work at how that information can be used later on. So by digitating crop trials, you're able to use and cross-compare against trials that have been done previously in a way that hasn't been seen before. And we'd like to do so at scale. We've done some in a number of breeding centres, but not at the scale that Bayer would be able to provide.
All right. So your presentation triggered a lot of questions. Unfortunately, for a matter of time, we cannot address all of them. Charles, you were presenting Fotenix. Thanks a lot for the pitch and the Q&A session. Now, let's meet Angela, who will introduce her startup called FungiAlert. Angela, are you ready?
Hi, Nelly. Before I start, I do want to, well, first of all, thank everyone to the opportunity, but I do want to highlight that unfortunately there seems to have been some compatibility issues with the slides I share, and unfortunately you will be seeing slides with some formatting errors, but I hope that doesn't affect my pitch. And now I'm ready to go.
Okay, so your 6 minutes start right now.
Sure. Can we change to the next slide, please? So, hi, I'm Angela de Manzanos. I'm the CTO and co-founder of FungiAlert. FungiAlert was founded back in 2015 by Carrie and myself while we were completing our PhDs in crop protection and sustainability at Imperial College London. Shortly after, we received some investment and we moved to Roehampton Research Centre. In 2018, we started our field trials, and we also welcomed 3 scientists with PhDs and postdoctoral experience in plant science. Next slide, please. Our patented technology, the Spore Sense, provides unique information about the soil microbial communities. Within the Spore Sense, generally there is a gel that contains a combination of chemicals and filters. So when it's placed in the soil or in the water, these chemicals mimic the root exudate, and it will attract the microbes that are normally attracted to the root zone, and it captures a sample of those microbes that are active and dominant in the soil. The Spore Sense then is sent back to our laboratory where we do species identification analysis, and we provide a very easy to read report. Next slide, please. Our Spore Sense microbial analysis is better than other techniques because our Spore Sense will only detect or will only give information about the microbes that are active and dominant in the soil. Other methods require you to send a soil sample to a laboratory. So the moment the soil leaves the field, basically you are taking the microbes out of their environment, and then in the laboratory they carry out isolation techniques, and ultimately you will obtain information about what microbes are active and dominant in laboratory conditions. Other methods that imply the DNA extraction from the soil and then the identification by, for instance, next generation sequencing will also detect the DNA from microbes that are latent or dead. Because our species identification analysis is carried out using Sanger sequencing, we're able to identify any microbe that has been captured by the Spore Sense, and not only to the species level, but sometimes even to the strain level. We also detect active pathogens in the soil before visual symptoms occur. Our current Spore Sense has been designed for the identification of fungi and mycetes, We do have one for bacteria, and we are in the development for one for nematodes. Next slide, please. We provide a report that is very easy to read, and this is because we characterize the microbes that we identify as pathogenic for the crop where the spore sense was placed, or with unknown activity. But interestingly, many times we do identify beneficial microbes like ectomycorrhiza or microbes that have potential as biofertilizer or biostimulant. Also, we do detect natural competitors of pathogens or biocontrol candidates. Next slide, please. Since 2018, we have carried out over 1,300 analyses worldwide in crops ranging from row crops to specialty crops, including hydroponics. And in this analysis, we have validated the use cases of our technology as a soil health analysis tool for the early detection of pathogens in the soil, also as a tool to study the modes of action or the impact of crop protection inputs on the soil microbial community. But very interestingly, our spore sensor also allows us to collect microbes from soils worldwide. So we have gathered a very interesting microbial collection. Next slide, please. FungiAlert has the potential to reduce the environmental impact of agricultural inputs. And this is because our spore sense microbial screening can guide the precision application of fungicides, reducing the carbon footprint associated to crop protection inputs. Also, as I said, we do have a very interesting microbial library, and we actually have identified potential biofertilizers. These microbial biofertilizers could replace or contribute synthetic fertilizers and reduce the carbon footprint associated to synthetic fertilizers. In addition, our microbial spore sense screening also can guide a smarter land reutilization and avoid unnecessary rotations. With the spore sense, we can also study what microbes are key to increase the carbon sequestration capacity of our soils and what farming practices can be done to promote the presence of these microbes. In addition, with our spore sense, we can facilitate organic farming because we can alert the presence of diseases in the soil, and also we can discover microbes that can be developed into products for organic inputs. Next slide, please.
1 minute.
There are many ways in which FungiAlert and Bayer can collaborate together, but here I have identified some examples. For instance, our spore sense data could be incorporated with data from the Climate Field View platform, and together we could develop robust models for disease forecast. In addition, in order to have a holistic view of how agricultural inputs actually affect the environment, we do need to understand what is the impact of these inputs on the soil microbial community. The spore sense can definitely provide with this information. As I said earlier, our spore sense is a tool to discover new microbes. So together, Bayer and FungiAlert, we can develop superior microbial products. And finally, the spore sense can also be incorporated into breeding programs to develop varieties that are resistant to soil pathogens, or also to develop varieties that interact or have better relationships with the soil microbial community. And the next slide, please. Thank you for your attention. I hope that we have the opportunity to work together soon, and we can help crops produce more with less using soil microbes.
Thank you, Angela. Now let's address the questions from the judges. So what makes your product stand out of the market, and how would you invest funding from Bayer to improve it?
Sure, that's a great question. So our product is patented. We do have 3 patents around our product. Basically, it is the combination of mimicking the root exudate and the process of placing it in the soil in order to discover and sample active microbes that are in the field is better than other soil microbial analysis, as I highlighted earlier in my presentation. How we will invest Bayer's funding in developing the technology. Well, the bacteria sensor has been developed. We do need, or we are actually fundraising in order to adapt our laboratory to be able to work with soil bacteria. And also the nematode sensor is in development at the moment. So definitely with Bayer, passion, if that was the case, we could definitely develop these products further. Also, we do have a very interesting microbial library, and we have characterized some of our microbial candidates. So with Bayer's collaboration, we could actually develop this very interesting microbial candidates into biological science.
Okay, so by when will the one from the nematodes detection be fully developed?
Very good question as well. So as I said, we are currently fundraising. So our fundraising strategy is definitely going to impact the development of our products. We have done proof of concept studies in our laboratory, and I feel that if the fundraising goes as we plan, we will be able to have an MSense in the fields before the end of 2021.
Okay. How do you know all of the active microbes are attracted to your compound? Isn't there strain-specific preference?
Yeah, that's a good point as well. So during these years of R&D, we have developed a composition that allows us to detect the microbes that are more important for growers in terms of soil pathogens. It is true that we can adapt the composition to sample different type of microbes. And it is true as well that we are always doing R&D and optimizing the composition of our spore cells in order to basically maximize the amount of species that we are able to detect. But yeah, definitely there is a preference of species that are captured by our spore cells.
Okay. Can your system be used to predict best crop rotations?
Exactly, and that was one of the points that I wanted to highlight earlier. Knowing what pathogens are present in the soil can guide what crop to plant afterwards. And on the other way around, maybe growers do change or do plant rotations because they think they have built a heavy presence of diseases in the soils, and maybe that is not the case. Also, with the spore cells, you can maybe analyze if your soils are actually very healthy, and you can plant whatever you want in your soil. So definitely, it has been used to guide the planting for the following season, and that is one of the applications that we envision as well.
Okay. Your method seems to work only for culturable bacteria and fungi, to the understanding of the audience. How does it differentiate from metagenome analysis?
Yeah. So I have to say that we have actually identified many fungi that have been classified as unculturable before. So this unculturable term, I think that I will put it into brackets because we have identified some that have been classified as unculturable. I said that it differentiates from metagenomics from a soil sample, basically because if you extract the DNA from the soil sample, the DNA from dead microbes and from microbes that are latent is going to be there as well, because the DNA from dead microbes can remain stable in the soil up to decades. So by doing next generation sequencing, you are going to be identifying microbes that are not active and dominant in the field. Our spore sense is only going to be detecting those microbes that are alive and active. Also, by doing next generation sequencing, you do obtain a lot of information, but it's more generic. You obtain information to the genus level. For instance, Finalists. With doing Sanger sequencing, we are able to identify to the species level. And there are species of Pythium that are pathogenic, but there are species of Pythium that are also beneficial microbes. So that is why it's so important to differentiate to the species level, not only to the general level.
Okay, thank you, Angela. You were presenting FungiAlert. I think we also received a lot of questions for your presentation. Your startup triggers a lot of interest, so that's good news. Again, we will store these questions and make sure that Angela have a chance to address them after the event.
Thank you, Nelly.
Thank you, Angela. Now it's time to hear about a startup called Kreatis, and it will be introduced by Paul. Paul, if you are with us.
Yes, I am.
Perfect. Okay, so I have to say that your 6 minutes starts just right now.
Okay, thank you. So my name is Paul Thomas. I'm the founder of Kreatis. And first of all, I'd like to thank Biocrop Science for giving me this opportunity to present Kreatis in this challenge and for your interest in our company. So why choose Kreatis? Well, first, Kreatis was founded in 2014. It's a unique and private research and development company specializing in silico models, predicting impact on human health and the environment. And the models that we use are a substitute for wet lab studies. They can be used to replace experimentation with confident predictions. Why do we exist? Our motivation, what gets us all up in the morning, is our desire to understand mechanistic interactions between chemicals and biological matrices. And we use this understanding then to quantify the impact on human health and the environment. What differentiates us from our in silico modeling competitors? Well, first, it's our aim for high accuracy. We're about data quality and not about data quantity. In fact, we reject probably 50% of the studies that we see because everything is validated in-house. We also have original approaches, and I'll talk about some of those later, such as the MECO, or mechanism of action approach. We also have dataset approaches, which are original. We use good modeling practice. Good modeling practice is a little bit like good laboratory practice, which is used in wet labs. We have a multidisciplinary group, and we work as a team, as a series of ecotoxicologists, toxicologists, chemists, and developers. And we also like to maintain a strong client-team interaction, so we provide support to our clients. Next slide, please. So this is the structure of our toolbox, which we call iSafeRat. I'll go starting at the top and going around clockwise around the structure. So we see at the top, we start with a chemical structure, which is input into our iSafeRat toolbox as a SMILES code. And from there, we determine critical physico-chemical parameters, notably octanol water partition coefficient, and also solubility, for example, which are generated from the chemical structure. These are very important because they can then serve as what we call descriptors for determining the toxicity of the compounds. The next point, and this is where the magic happens, it then passes into our MECO scheme, which is a proprietary scheme developed in-house. And MECO is mechanisms of toxic action. So we use that to quantify the toxicity. We currently have 26 pathways today, and we think there are maybe 50 or so pathways in total for organic chemicals. And MECO looks at the structure of these chemicals to decide what the adverse outcome pathways are. These are called AOPs, and the pathways that they then may follow when we get to the toxicity side. So we then move to the toxicity determination. So we have aquatic ecotoxicity and human health. On the aquatic ecotoxicity, we follow regulatory study prediction or outcomes. So for algae, for Daphnia, for fish, and also for microorganisms. And we try to do this for acute and also for chronic endpoints. And the interesting area also within aquatic toxicity is I believe we're unique in having determined a thermodynamic methodology for looking at toxicity for mixtures. Then there's the human health side, which we're still working on. We only have a few models at this stage. We have skin irritation and eye irritation models, dermal absorption, and we also have a model for skin sensitization. And a major area that we've been working on now seriously for the last 2 years has been on the endocrine disruption side. So we have a collection of models using both 2D and what we call docking. And the idea is to determine potential for substances to be endocrine disruptors or also substances not to be endocrine disruptors. And the 2D docking is a structural alert, which uses the same methodology as we see with the MECOAs. The docking system is a 3D methodology, which is much more complex. Next slide, please. So why is iSafeRat a sustainable solution? There are advantages and in silico tools which are multiple. First, they're ethical, they're rapid, and they're a low-cost alternative to wet lab studies. And our models have the added benefit of being highly accurate. they can screen out undesirable chemicals before they reach a higher level in the R&D chain. And this can save time and money and also reduces risks when it comes to eco-conception. And the good thing is with iSafeRat is it can only get better as in silico techniques evolve. Next slide, please.
1 minute.
Okay. What does Kreatis intend to do next? Well, we don't have all the answers yet. There are millions of possible chemicals out there to explore. And we understand mechanisms for tens of thousands of molecules, but we need to improve in some areas, and notably in the area of plant protection products. We want to move to an even higher level of endocrine disruption detection than 2D or docking. We want to move to molecular dynamics simulations. And so far, most endocrine disruption research has been on humans and vertebrates. We also are interested in looking at non-vertebrate animals too. So finally, we have an ambition to model reprotoxicity, and this is a subject which is a bit related sometimes to endocrine disruption too. Next slide, please. So what are we asking from Bayer Crop Science? Well, first, we're asking Bayer to be our client and to use our tools, and ideally be a long-term partner with us in a research project. We really need data. All our work is based on quality data, and to have access to your data would boost our models in areas that are less strong today, and notably in the area of crop protection. And finally, we're a small startup, so some help with investment in some of the expensive areas of software and hardware could possibly help us to create our models and make them go faster in some areas. So thank you very much for your attention, and I look forward to receiving your questions.
Thank you, Paul. Okay, let's start with some questions from the jury. So, concerning your plans for a reproductive tox model, what type of data will you use? And are you generating this data, or are you expecting this data from partners only?
Right, it's a great question. I think there is already a lot of reprotoxicity data which have been, thanks to REACH, there are enormous amounts of data which are actually already out there. But we still, and most of it's come from in vivo studies, which were carried out in the past. Obviously, our aim is to minimize in vivo studies in the future. So we will have the opportunities to be able to move forward with reprotoxicity studies which already exist. However, as I said, one of the areas that we need to look at are these multiple mechanisms of action. And we know, as I said, there are at least 26 and possibly 50 different mechanisms of action. So we need high quality reprotoxicity data across all those areas. It's quite likely that in vivo studies don't exist for all types of groups and in a way that would allow us to be able to create a model, a functional model for each MECOA. And there, I think there are in vitro studies that could be used, which could certainly support that data. And we could certainly move ahead without using animal models. So there would be no need to do further in vivo models. Sorry, in vivo studies.
Okay. How do you in silico models translate in vivo?
Well, in vivo, the idea is that they translate perfectly. What we use as our models are experimental studies which have come determining intrinsic properties of toxicity and ecotoxicity. So we're using the standardized OECD methodologies which are there to measure intrinsic properties of toxicity. So they relate very closely to an outcome that you would get if you ran that regulatory study. The regulatory studies are classically OECD guideline studies or equivalent.
Okay, so what is your definition of accuracy if you talk about how accuracy tools?
Right. What we try to achieve with accuracy is an accuracy which is similar to what we would expect from a valid experimental study. I'll give you an example. If you run a fish study, for example, in an experimental laboratory using the same technician 2 weeks in a row, you would expect to get nothing more than a factor of 2 difference between the 2 study results. And therefore, we consider that within a factor of 2, for that specific type of result, that would be considered an accurate result. So if our estimation was within a factor of 2 of an experimental study, we would consider that to be accurate. And that each endpoint has, of course, its own definition of accuracy, but we consider that we need to be as accurate as we could reasonably get from a repeatable laboratory experiment.
Okay, maybe a last question. What other tox endpoints can you address currently?
Well, at this stage, we can't cover other tox endpoints than those that we've put on the list, but we are interested in moving forward with repeat dose toxicity, LD50, and as I said, reprotoxicity is something that particularly interests us. And we're also, we've not finished yet this long journey with regards to endocrine disruption. And there is still some way to go. We've made a lot of progress, and we are moving now forward towards the final stages of the 3D models. But the molecular dynamics simulations is really a next step that we have to embark upon.
Okay, thank you, Paul. That was the last question. You were presenting Kreatis, so thank you for the pitch, and we received some questions for you too. So we will make sure that you have a chance to answer them after the event.
It'll be my pleasure. Thank you.
Okay, so now let's move to a startup called Walrus, and it's Scholl who will introduce this great startup. Are you ready, Scholl?
Yes, thank you, Nelly.
Okay, so your 6 minutes start just right now.
Hello, everyone. I'm Shaul Zavian, co-founder of Walrus. We are a young startup company, and our product is an autonomous system for indoor farming using smart microdrones and AI for early detection of pests and diseases. Let's start with the problem. Indoor farmers fail to prevent pests and diseases. Why? Because of late detection, caused by 4 reasons: lack of time, lack of employees, lack of treatment, and lack of knowledge. As a result of late detection, pests and diseases spread throughout the greenhouse, forcing farmers to use lots of unhealthy pesticides, which are catastrophic to the environment, worth tens of thousands of dollars annually. The consequences, and I really hope you will remember the number, are annually yield loss of up to 30%. Yes, 30% loss. Now, in the next slide, I want to show you a graph taken from a research done in a university from Sweden. The graph will explain the difference between early detection and late detection of a disease called powdery mildew. In the x-axis, as you can see, is the days starting from the detection day 0. In the y-axis, you can see the severity of the disease in percentage. The red graph show a crop untreated, and the green graph show a crop treated from day 0, the detection day. You can see that in the first 2 weeks, the two graphs behaves almost the same. But then the untreated graph jump up drastically, while the treated graph stay almost the same. This is exactly what we want to achieve. Our vision is to help every indoor farmer in the world to detect pests and diseases early enough and apply the right treatment, just like in the green graph. And remember, there is a 30% loss. Next slide, please. And this is exactly what every farmer wants to see: green and healthy crops. With the help of our system, we can save time, employees, and give access to missing knowledge while reducing reaction time and increase yield. Next slide, please. How does it work? We use autonomous drones for scanning the plants, just like in the upper picture. Then we diagnose the picture using our AI algorithm, just like in the bottom ones. Once done, the farmer will get recommendation for the right treatment from the cell phone app and can react immediately. The benefits will be huge. Reduce reaction time to almost 0. Save time and employees. With the drone, we can enable a per plant detection and precision application of treatments, increase yield, cut losses and pesticides expenses by up to 70%. And finally, we can give access to missing knowledge. Next slide, please. Sustainability. One of our system's greatest benefits is reducing pesticides use. Researchers show that pesticides use can be reduced by the use of precision application by up to 70%, which is really big number. Per plant detection and precision application. Once pests and diseases detected early enough, treatment application can be used at the exact point inside the greenhouse, or maybe just take out the infected plant without any further treatment. Last, by promoting biological products through Walrus system, we can help indoor farmers sustain healthy produce, reduce environmental contamination, and reduce pest resistance. Next slide, please. Where we got so far? We finished POC and deployed 3 stationary systems, which are helping farmers as we speak. Our next milestone will be to raise funds to develop and deploy 3 drone prototypes and then head to mass production. And as you can see, this is a really big market. It's almost 6 million hectares around the world. Next slide, please. Cooperation with Bayer. We see Walrus and Bayer doing lots of things together. For instance, customer acquisition. As an international organization, Bayer can help us with customers acquisition around the globe. We can boost sales. Walrus can help Bayer to promote its services and products to farmers through our app. Knowledge, we can share knowledge about pests and diseases. But finally, Walrus could be Bayer's window to every indoor farm in the world. Next slide.
1 minute.
I want to thank you all for taking the time to listen to me. If you want to keep track of our progress, you can visit our website, walrus.ag, and I'll be happy to answer questions now.
Thank you, Shaul. So let's start with the questions from the jury. What makes your product stand out in the market, and how would you invest funding from Bayer to improve it?
Okay, so first of all, this product was born from my experience as a farmer. I worked as a farmer for 10 years, fighting those pests and diseases every day, every year, every crop, every month. And basically, what we want to achieve with the money is to develop the prototype of the drone, filming the crops and doing it all autonomously. And by autonomously, what I mean is that the drone will dock inside the greenhouse and will operate by itself by a pre-scheduled by the farmer. And this is basically our greatest advantage of all the others competition, because we do it all autonomously and directly to the farmer.
Okay. Regarding the investment, maybe?
Yes, we will invest the money in developing the prototype of the drone, because right now we use only stationary systems and we want to move on to the drone. So we need money to develop it and to put it to use and test it.
Okay. Can you remind us how early can your technology detect the emerging problem?
Well, basically, according to our experience with the stationary camera, we can do it by a matter of seconds. Once the picture taken from the greenhouse and sent to our server, diagnosed, and the algorithm decided that if he diagnosed pests or diseases, he sent notification or alerts to the farmer through our app, and it all happened in a matter of seconds.
Can you explain your comparison of costs for value with other companies?
Yes, we want to deploy one system per 2 hectares or 5 acres, depends on which country you live. And basically, it shouldn't be more than hiring a private scouter or inspector for doing the job yearly. But what we can give is 100% coverage if I want to differentiate us from the personal scouter, which will come only once a week and do a sample of the crop. And we can do 100% coverage and every day.
Okay. How do you avoid greenhouse obstacles with your drones?
Basically, our plan is to cover the drone and to put it inside a cage, which is very, very light, in a sphere, like a cage, like a sphere, which the air can go out and in. And it basically would operate like an iRobot if you got it in your house. Once it will bump the leaves or the crop, it won't fall. It won't do any harm to the crop, but instead it will just know that it bumped into something and he will get back to his truck. So no damage, not to crops, not to humans, and not to the drone.
Okay. Is the processing of information and recommendations expected to work in real time?
Yes, it all will happen in a matter of seconds. Once the drone film the picture, it will send it to the server. The server will diagnose those pictures, and with the help of our computer vision algorithm, we can decide if we detected something. If the answer is yes, we send it immediately to the farmer, and it will all happen in seconds. No delay, no 3 hours or 1 hour. It will all happen very fast. We do it right now, but with stationary cameras.
Okay, maybe a last question. We have a lot from the audience, so thanks a lot for sharing your questions. Can your solution detect also hidden pests and diseases, like, for example, dwelling on the underside of the leaves?
Once it gets to pests on the bottom of the leaves, what we do is looking for the spots which they make. Like if I take, for example, the red mites, they are making those yellow spots on the leaf. Once we detected those spots, and at the stage that they are really tiny, and the red mites didn't consolidate yet inside the greenhouse, we immediately can detect, and we can tell if it's red mite or downy mildew or powdery mildew, for example. This is how good is our algorithm right now. So this is how we do it.
All right, well, thank you. Time is up for the questions now.
Thank you, Nelly.
You were presenting Walrus, so thanks a lot for your presentation. You had a lot of questions, and we will make sure that you have them after the event. It's now time for me to hand over to Simon and to thank you for your attention.
Thank you. Thank you, Nelly. And first of all, I think you'll share with me, that was absolutely fantastic. And I'd like to thank each of the presenters in order. So Yoav, Margaret, Charles, Angela, Paul and Shaul at the end. Well done to all of you. I mean, I've had to take a seat. It's been overwhelming. It's been absolutely fantastic. So I'm taking a seat here in the plants. And there's a couple of messages I've got for you now. So the first one is, you can see on your screen now, you can see a QR code and you can see an address to a website with a code underneath it. So those of you that are already got your iPhones out, you can scan that code. And then it's a chance for you now to vote for your favorite startup that you've seen today. And you'll get a choice of up to 2. So you can choose up to 2 different startups from the list by logging into that website. I'm going to ask Nelly if she can put the address and the code into the chat, or if I can ask Kyle to do it. Put the address and the code into the chat, just to make sure you've got it. And I'll leave that on for a little bit longer, because I want to take this moment not only to give thanks and applause and praise to all of the startups, so thank you, that was absolutely fantastic. The crowd here are going wild. I also want to take a quick moment to pause and to thank the tech team. So the guys behind the scenes that you can't see today, they've been fantastic. So Carl and Matthew in the US, and we've got Arno with us here today, and everyone else that's working around them, and the team in the Life Hub as well. I want to thank immensely for everything. So everything's running so smoothly because of their hard work. So really great thanks to you guys. I've got a couple of other messages to do now. So what's going to happen is, we've seen all the pitches. You guys are going to have a chance to vote. And also our judges are going to move into their deliberation room, and they're going to start discussing in order to choose a finalist, in order to choose a winner that will present at the end of the event. While they're doing that, I think it's a great opportunity for us to introduce Vincent and Chloe from Hello Tomorrow, who are with us today in the Life Hub. They're going to say a few words about Hello Tomorrow, about the Global Summit, which is coming up. and I'm really looking forward to hearing what they've got to say. So don't go away, and stay with us until the end so you can hear the winners. And in the meantime, let's have a quick listen to Vincent and Chloe from Hello Tomorrow. So thank you very much.
Thank you, Simon. Thank you. Well, before presenting Hello Tomorrow, we'd like to introduce ourselves. So Chloe Khalifa, our deep tech innovation consultant at Hello Tomorrow, and myself, Vincent Durand, head of startup corporate programs. So yeah, we're both thrilled to be here today at the Live Hub Lyon. It's a great pleasure to be here and also to see what we did this last week with Simon, with Phil, with Philippe, with many others, come to an end in the most inspiring way. So thank you and congratulations to the 6 finalists. When I say end, maybe it's not the right choice of word as well, because I'm pretty sure that these will lead to many great, inspiring, and impactful collaboration between biocrop science and these startups. So that's really great. Next slide, please.
Yes, so our mission at Hello Tomorrow is mostly to unlock the potential and even power of emerging technologies. They're often referred to as deep tech, and they will help us solve our toughest challenges. Now, when I say deep tech, it can go from artificial intelligence to synthetic biology to even quantum computing. What they all have in common is the fact that they all will have more research and development needs than a regular startup, will need more investment, often have a longer time to market, but they truly have the potential to transform our industries.
Next slide. Thank you. So when she says unlocking the power of deep tech, what does it mean for us? At Hello Tomorrow, we have 4 main activities for that. First, we scout the best, most innovative startup deep tech solutions in the world through competitions like this one or like our annual global challenge. And Chloe will explain it in a few minutes. Then we connect all these great startups with other key stakeholders, being industry leaders, investors, incubators, universities, all of these key stakeholders of these ecosystems through global conferences and events offline and online. And our flagship event is called the Global Summit, and I will also present it in a few minutes. Building on these 2 pillars, events and competitions, what we do also is we analyze trends. We issue every year reports on these on deep tech, on innovation trends, and position ourselves as well as opinion leaders in deep tech. And lastly, our main activity for Chloe and I here is consulting services that we provide to large companies like biocrop science. We help them understand and discover deep tech. We help them scout the right startups and solutions for them, and we help them accelerate their collaborations together. Next slide, please. And to do so, we are fortunate enough to have a great international community behind us. We are present in 7 countries in the world, including Hungary, Turkey, Japan, Singapore. And also we can count on 72 representatives in many countries, I think 30 countries in the world. And we never thank them enough for everything they do, bringing great solutions, great deep tech startups in our network as well. Next slide, please. When I say network, today our network has deep roots in all major innovation hubs around the globe. It consists of 6,000 deep tech startups, basically the best startup that we gather from our global challenges. And yes, Chloe will talk about that in a moment. I think it's next slide. Hundreds of industry leaders, corporates, and investors, but also 250 what we call ecosystem partners, incubators, universities, research institutes, and centers, and all of them gathering all of these great ideas in the Hello Tomorrow network. And yes, this strong and vibrant network, I wanted to add as well, is that this is the network that allows us to do these competitions and allows us to do a challenge like the one we're doing today with biocrop science. So thank you again for them. Next slide, please, for the global challenge.
Yes, absolutely. So the global challenge, like Vincent said, is one of our pillars. We've been having this international deep tech startup competition since 2014, and this edition was quite successful. We've had 5,000 applications, and they cover over 100, well, not over, but just 128 countries, which is quite extensive. Out of those 5,000 applications, we have selected 750 of what we consider to be the best technologies for deep tech. So we call them deep tech pioneers, and they will all have the opportunity and are all invited to our annual event, which is the Global Summit. Vincent will talk about it in a couple of minutes. With the Global Summit, what they get is a chance to meet our ecosystem, key stakeholders such as investors and corporates. Next slide, please. Thank you. So out of those 750 deep tech pioneers that we've had this year, 80 of them will pitch live during the Global Summit. So why 80? We have 14 different tracks covering from AI to new space to energy, drug discovery, and of course, food and agriculture, which we're very happy that BioCrop Science is a partner of this year. And they will all, during those 5 days, showcase their solution. And eventually, we will end up having to vote for our global challenge winner.
Exactly. Next slide for that, please. Each year, we vote for the global winner cross category. And here you can see the the 5 winners of the global challenge, and you can see the wide variety of solutions we have here in 2019. Rexel assessing in real time drugs and in order to fight fakes. We have Sati producing biodegradable sanitary pads for women, for millions of women around the globe. We have also biocarbon engineering, now called Dendra Systems, allowing mass reforestation with drones. And they actually met one of their investors at the Global Summit, the CEO and co-founder of Parrot, the drone company. So next slide, please. Global Summit. We talked about the Global Summit, the end of our annual Global Challenge. This year, it will be held online between the 16th and the 20th of November. We usually do it offline in Paris in March, gathering 3,000 to 4,000 people. And yes, we know this year it won't be like this. This year it's different. Many things are different this year, but different doesn't mean that it's not going to be great. And on next slide, you will see what we designed for this event. So these 5 days now will end with a 5th transverse day, but all the fourth day will be focused on one broad theme. Each day you'll have the opportunity to discover new technologies, to attend startup pitches, to listen to compelling fireside chats, to ask your questions, to also listen to inspiring keynotes. And also you'll have the opportunity to network with the other attendees using our tool that we have linked to this Global Summit. If you look at day 1 on November 16th, focused on humans, so healthcare, well-being, medical biotech, I guess an unmissable day considering the current pandemic. Day 2 will be focused on machines and their relations with humans, meaning AI and robotics. Spaces for day 3, bringing closer together of societies, but also outer space. And maybe the day that you are most interested in, the 4th day on planet, with the future of energy, environment, and obviously food and agriculture. So next slide, please, for the food and agriculture.
Absolutely. So I mentioned the fact that there are 14 tracks. One of them is food and agriculture. We're not going to present you the finalists. They'll do a much better job than us on the 4th day, which will be on November 19th. This is just to highlight the fact that the technologies are quite broad all around food and agriculture. And once again, BioCrop Science, we're pretty thrilled that BioCrop Science is our partner this year for this track. Next slide, please. And like Vincent mentioned, on top of the startup pitches, you will also get to witness keynotes, but also fireside chats. So regarding the food and agriculture program, it's not 100% definitive yet, but we do know that you will be able to learn about about securing our food systems, but also the status and challenges with lab culture meat with Ira Van Allen, who's the CEO of Kind Earth Tech.
Yes, and on next slide, almost the last one, before joining us at the Global Summit, and now that you've seen these awesome startup pitches and I guess our presentation, you will join to our virtual Global Summit. Please take a look at our publicly available reports and trend analysis that you will find online on our website. And last slide, please. Thank you again very much for your attention. Thank you to Bayer for trusting us for this challenge and for making this event possible. Thank you and congratulations to the finalists and all the applicants. You are all bringing much needed and valuable innovation to the table. So thank you, please continue. And I will give the mic now to Phil, talking to us about open innovation in Bayer. And it's not just about Hello Tomorrow, I guess, right, Phil?
Yes, thank you, Vincent. Thank you, Chloe. I really appreciate the opportunity to hear about Hello Tomorrow and the organization at large. Hello, everybody on the call. My name is Phil Taylor, and I'm the Open Innovation Lead within the Strategic Partnerships groups at BioCrop Science. And it's my pleasure to spend a few minutes now just talking a little bit about the frame of how we think about the innovation that's needed in agriculture and how we think about how open innovation plays a key part in that. So if we can jump to the next slide. And maybe the next one, and maybe the next one. It's no surprise to anyone on the call here today that, and you've heard it in all the pitches we've heard from the companies this morning, that farmers need innovation. We need to think about that innovation in the context of growing enough, but also doing it in a better and more sustainable way. And at Bayer, with our mission of feeding the world without starving the planet, we have a real role to play in driving agricultural innovations, being part of the solution to the hunger challenges in our world, to the climate change challenges in our world, and thinking about how to use decision-making, but also the biological and the chemical tools at our disposal to really amplify the impact of agriculture and drive innovation in a very, very broad sense. The next slide, if you don't mind jumping forward, is really just a sort of a description of really how we think about that and how we think about doing it. And you've heard today a number of different technologies and different ways that can impact this sort of landscape. But we really think about productivity as the combination of the right genetics and the right tools that we have in our genetic and crop protection toolbox, with the integration with the environment, understanding what is the right material to plant or treat or work with in a particular environment that is ever changing. And coupling that with the farming practices that our pharma customers are aware of and driving in their own operations. Farmers make over 40 independent decisions every year about what to plant, when to treat, when to harvest, how to harvest, and helping them put all that together in a singular package and a singular sort of set of tools is really the mission of how we think about bringing innovation into agriculture. So by combining our leading products and also the combination of those products with their digital offerings to help make decisions and help support analysis on farm, we really are driving the concept of tailored solutions to support our pharma customers. And so it's not just about a bag of seed or a crop protection chemistry. It's really about understanding how you bring those innovations together, how you have those predictive tools, many of which you've heard about this morning, or how you have that analysis that allows you to know when the right time to make the right decision for your operation. Another example is on the digital side with the Seed Advisor, where we're combining the largest genetics library in the industry with all the performance data that we can bring together to really understand the best performance and the best opportunities for our pharma customers. So this in and of itself is a pretty exciting sort of landscape for agriculture right now. But when we think about it within Bayer and certainly within our team and in my group, the real excitement is how we think about this from an open innovation perspective. Next slide, please. We know, even though we're very fortunate enough to have one of the largest, ag tech R&D organizations on the planet, we can't do this alone. And so we leverage a number of open innovation sort of opportunities and vehicles to really drive acceleration. And as Bernie said in his remarks at the beginning, I couldn't agree more, to help us think about things through different lenses, help us think about things through different perspectives. So whether it's key partnerships with universities and research institutions, engaging in sponsored research where we're leveraging expertise for organizations that have a specialty where we need some support, to crowdsourcing type opportunities, helping us find new startups and new academic partners to work with, much like this partnership you're hearing today with Hello Tomorrow. You know, we try to find as many ways as we can to meet innovation where it is and support its need depending on what the innovator, no matter where they are in the world, needs in terms of their support. Another angle on this is also how we think about innovation. You're seeing today that the event is coming to you semi-live, I suppose we're calling it, from our Life Hub in Lyon. And our Life Hub network of 7 or 8 locations globally are physical presences for us to drive open innovation, whether that's through networking or co-creation or incubation of startups. We want to have that footprint out there in the different ecosystems to be driving and accelerating innovation. And then at the same time, we're very focused on leveraging classical collaborations, be they licenses or R&D partnerships with startups, some of which are called out on the slide here, all the way through to our venture capital organization with the Leaps team, Leaps within Bayer, making those venture capital investments to accelerate startups. And so you'll see we have a number of tools in our toolbox. We love to think about this as a very diverse mix that allows us to meet innovation where it needs support and accelerate it. And you know, I would, if you see something on here that resonates, or you know you're interested to learn more about it, I'd love to connect. Maybe just a couple of examples on the next slide of some things that are going on right now within the team. Obviously, you're here today as part of the challenge we did with Hello Tomorrow and Vincent and Chloe that you just heard from. And we're super excited about these pitches. I can't wait to hear who the winners are. But in the same time, you know, we're also in the middle of running our very popular Grants for Ag program, where we give pre-competitive funding, small euro amounts around 10 to 15,000 euros, to accelerate early innovations through that first experiment, through that first proof of concept that they need to execute to understand if their innovation is applicable or relevant and useful for agriculture at large. And we do this in a very pre-competitive way with no rights or restrictions or reporting or requirements or anything like that. It's really just about making sure that we accelerate the science. So if that's a program that's of interest to you, we can certainly share some more details. But as I say, the program's going on now, closes at the end of next week. And with that, I think going to the last slide, just like to acknowledge, you know, Simon, who you've seen. This is what he looks like without his mask on. You know, who's been instrumental in working with Hello Tomorrow to pull off today's event. And we would love to connect with you if you have an idea that fits the frame of what we've talked about here today, or any of the other sort of open innovation approaches that we're taking within biocrop science. We would love to connect. Our Twitter handles and LinkedIn links are there on the slide. So thank you for your attention, and I think I turn it back to Catherine.
Yes, back to Lyon. Thank you, Phil. That's really a pleasure to welcome you in this location. So the Life Hub, this place that you have seen all along the presentation, we have tried to, even if we are in virtual, to give you a feeling of this location, the real places through the different person talking. So this place is really meant both for innovation and dialogue. This is where we welcome a certain number of visitors. And the advantage is that it's back-to-back to the research center. On these doors that you don't see here, you enter the site and you have 200 researchers here. So what is great is that it's really a perfect way to have this networking and this open innovation in a win-win composition. And you really expose all our researchers to the new technologies. So the research that is done in the research center, it takes about 10 to 12 years to come onto the market. So we need to be really listening to our customers, to our to the population, really to define and think in advance to the future solution. That's why those events with the startups are so interesting. So I'm having the clock running, but I'm waiting also for the judges to come back with the final decision on who will be the winner. I'm sure that you are as impatient as I am, but it seems that it's not totally ready yet. They are still debating, apparently. So I can maybe take some words about what we do in this location for the different visitors. So we have a culture of transparency. We really want to explain what our researchers are doing and how they work from the really early discovery of new active ingredients, for example, or it can be traits or seeds. So we try from this early discovery to explain all the research process until the development. This is really important to showcase also all the steps that a compound, for example, can have to fulfill before to be accepted and entitled to be sold in the market. So to do that, for example, and to explain what we are doing in terms of safety of our compounds, for example, we have an open lab where it has been refurbished recently, and this is really great to explain, for example, residue analysis processes and all that is related to those processes. This is about thousands of visitors that we welcome per year, and all our visitors are really, I think they really enjoy our transparency, the direct contact to the researchers too. So I'm seeing some people coming, so that's great. I think the deliberation just ended. So Bernard, Simon, I'm so happy to welcome you back to announce the winners. So
thank you, Catherine. So this was one of the most exciting sessions I've been through in the last year. Certainly something new to me, but because of the context, of course, but certainly great pitches from our presenters, really a nice job, and it offered a lot of discussion between our judges. Now, after all that discussion, we finally decided that Kreatis would be our preferred judge, and we should really applaud them. One of the reasons we chose Kreatis is the fact that we see a real opportunity in their proposal to actually improve much further the way we actually assess our product. We have a very significant amount of data that could go into modeling, and we see their approach as very futuristic and really in line with the public requirement to use less animal for testing. That is one of our aim, and we think that in there we can have an excellent partnership. So that's what we would go for. Maybe we'd like to have some feedback from Kreatis now.
Yeah. Paul, are you on the phone?
Yeah, hello. Well, thank you, really. To be honest, when I saw the quality of the finalists, I really didn't expect that we would win. There's so much promise out there. There's so many interesting opportunities, and so I've barely prepared anything really to answer to this. I will be happy to answer any further questions that came in during the presentation that I didn't get a chance to answer when I was being questioned at the end. And I'm really looking forward to moving ahead and working with Bayer in the near future with some really exciting, I think, really exciting potential projects that we can set up together. So really, thank you very much. It's been a really fantastic opportunity.
Pleasure.
Pleasure.
So that being said, it's as I said, it was difficult, but we had also another group of people were actually involved in assessing the situation and the pitches, and the public so voted, and the vote of the public is actually Agrosage, and it's very good because actually when I look at the balance of things, they offer the other side of what we were looking for, which is actually advanced digital approaches to actually detect pests and disease, but also possibility to actually reduce the load of chemical we can put in the environment while ensuring a good result. So that is really a good balance from that element. Maybe Agrosage want to say something.
Yes, it's in patch.
Yes, so thank you. Well done. Yoav from Agrosage, maybe you have a few words?
Yes, sorry, I was on mute. Thank you so much. This is very, very, very exciting being the choice of the crowd. Thank you, Hello Tomorrow, and thank you, Bayer. And we are very, very excited in doing the next steps with Bayer and finding the best solution for farmers with early detection of pests and diseases. So thank you very much all.
Now I'm back to you guys.
Yes, so I mean that's just been absolutely fantastic. Well done again to all the startups, to all the pitches. I think Catherine and I, we just have a few closing words, I think, to wrap the session up.
Yes, exactly. So I think you really saw the spirit of this place, but also the open innovation for Bayer. So it's really important to us, this win-win situation where really we can open our doors and explain our solutions and be transparent. And on the other hand, we really enjoy seeing the energy and the nice ideas from the entrepreneurs. It's clear, it was really visible through the pitches. So I think and hope you really saw that. So it's almost the end for today, but definitively not the end for us for open innovation. So we have, as Phil explained you, one running challenge at the moment. This is with AgConnect. No, hello, help me.
Hello, Grants for Ag with Grants.
Hello, Grants for Ag. Merci, Simon. Sorry for that. By mid of November, we will have also the Global Summit with Hello Tomorrow, as it was explained just before, and we really hope that you will take place into that. So they can even join us on stage, and that's really great because then you saw that we will have here a great event. And in March, next March, we will even welcome the European Data Science Elixir with a nice event also that is planned already to be virtual with a lot of nice talks on data-driven innovation in the agri-tech sector. So we also want you to participate. And for the next
Thank you, Catherine. And then just to wrap up, I've got a couple of notes. So, I mean, it's been a great 2 hours. We've run through a couple of topics. We've run through open innovation at Bayer. You've heard how important it is to us. We've run through the pitches from the startup, which was really a fantastic highlight. We're really pleased that we've been able to choose a winner from the fantastic 6 finalists in Kreatis. The company which Paul presented. And we're fantastically happy that Agrosage got about 43% of the vote from the public. So a real hats off and congratulations there to Agrosage. And we're going to finish this off now. I've thanked everyone. We've done the winner's announcement. And so really there's just one message for me left to give. And And I hope that you've had fun. I hope you've enjoyed yourself, and I hope you've learned something today. And I also hope that you'll join us on our journey together, so that we can shape the future of agriculture together in the future. So from my side, thank you very much for coming. It's been fantastic. Thank you to everyone here, and thank you to you all who took the time to watch us from home or from work. So thank you very much. Goodbye and see you soon.
Thank you, everybody.